Hello, everybody. I see a lot of you are coming in to the session. So I'll wait another minute until you are all in to start the programming. So first of all, I want to introduce myself before we go to our program today. My name is Francisca Elmer. I'm an independent researcher at the moment, and I co-organize Global Coral Reef Week. And with me helping to manage this session is Chelsea Council. She's also helping to organize Coral postdoc at in Hawaii. Today we plenary talks by Assistant Professor David Gill and by Dr. Brigid Tussenbrook. After the two talks, we will have time for questions. And if you have any questions to our presenters, please put them in the Q&A chat, which you can find at the bottom, and not that way it's easier for us to see the questions and your question will not get missed. Please use the chat to introduce yourself, to talk to each other. It's a great place to get to know the other people who are listening to this talk and to talk to each other while listening to the plenary talks. For the Q&A questions, you can upvote questions. So if there's a question you read, you can upvote it so people see that this is a question more people want to see answered. If you are having any technical difficulties and you don't see the chat exploding with the same difficulty, then probably it's only you who has this difficulty at this time, and it's due to your device or your internet. What you can do is you can log out and rejoin the session, or we are also live streaming this on YouTube. You can go over to our YouTube channel, Global Coral Reef Week, and watch it there. Or if really things go wrong, you can watch it later there as well. We're going to start off with two quick polls because we want to know who you are. We want to know where you're from. So our first poll is about where you are located right now. All right, I'll give you guys another 5 seconds or so to give your answers. And let's see. So we see we have a lot of North American and South America, like we did in the last section. Caribbean people have increased. So a lot of Caribbean people probably interested about the sargassum. And also a lot of people from Europe in this session. So now I would also like to know what sector you are working in. All right, so if you haven't done it yet, try to answer the question. We will close the poll in about 5 seconds. So very similar to the last few sessions we had, a lot of academics and students, but also quite a few people from government and conservation. Very cool. And a little bit more from education than before too. So without further ado, I would like to introduce to you our first speaker, Assistant Professor David Gill. David Gill studies marine coupled human natural systems. So he looks at marine management, and especially marine management in tropical coral reef systems. And he tries to find ways through research to get equitable and sustainable outcomes through management and conservation. And he is working at Duke University in North Carolina.
Hi, welcome. So do I just share the screen and start?
Yes.
Okay, all right. Well, thank you very much for the invitation and for hosting the exciting series. Can everyone see the PowerPoint screen? Yeah, okay. All right, so today I wanted to talk a little about marine protected areas, but taking a step back and thinking about how we understand and measure the impacts of any type of policy that we implement to protect coral reefs. So everyone knows about some of the major threats to reefs. And one of the big responses to that threat are marine protected areas. We've seen an explosion in the numbers and sizes of marine protected areas around the world, where we're reaching, probably getting closer to the original 10% target that's driven by many international agreements, such as the Aichi targets and now in the Sustainable Development Goals. But then when we think about, well, are these MPAs actually delivering the social and ecological benefits that they're designed to produce? And how do we go about measuring and monitoring marine protected areas? And when these approaches vary, but today I'm just going to talk about some of the ways that in the field, we're collecting primary data to understand what's happening inside on our coral reef and the MPA that we've invested so much work into designing and implementing. How do we go about measuring and seeing if it is effective or not? And then at the end, I'll talk about an example where we tried to assess the impacts of marine protected areas on coral reefs and other similar habitats. So when we ask about measuring impacts, what are we talking about? We're talking about the difference in outcomes that we see in observations that we can attribute to the intervention. We can say, yes, that this change that we have seen, it is because of this marine park. So we would need first to some kind of measure of change or difference in outcomes, whether it be before or after the intervention, or when inside or outside a protected area. And then if we want to do it well, we need to consider the other factors that cause changes in outcomes in coral reefs. There could be massive coral bleaching events. There could be a development happening nearby. And then that affects both the environment as well as the people who depend on it. And the three ways that we generally look at and monitor and measuring when we're doing reef monitoring is looking at differences in status and trends. And then I'll talk about impacts, which is getting a little more detailed. So let's start off by talking about some of the ways we do it spatially. We may, for example, be interested in looking at tourism income on a coral reef MPA. And we see that tourism income is higher, maybe like 5%. We think that we've done a good job. We're comparing MPA and non-MPA locations. Both sites are perhaps subject to broad scale impacts, such as if there is some form of pollution, it happened to both sites, or if there is some broad economic policy, both sites still have been equally affected. However, when we do these kind of spatial comparisons, we often don't account for some of the major differences. It could be that our MPA site is actually healthier in better conditions. Or in this case, or in another case, it could be that they do not account for the other activities that are happening currently. This was happened in a study on an MPA in Brazil, where they were looking at fisher income between MPA and non-MPA sites. And they did see higher fisher income in the communities near to the MPA, but they realized that that might have been because of differences in fishing techniques. And actually a site that was even further away, the income was a lot greater. And that was because that site had its own form of community-based management rules. Okay, so then let's talk about temporal comparisons. We may see, since we implemented our MPA, a 2% increase in coral cover. We are looking at trends and shifts as a result of intervention. We don't need to worry about differences between sites because we're considering the same locations. And then we see the change in the trend since the marine protected area. I may say, aha, we've got it. We've measured a 2% increase in coral cover. This is a great approach, but it often does not account for these other factors that contribute to change. What would have happened if before you did your monitoring, there was a storm, and instead of measuring MPA recovery, you're measuring storm recovery or other types of shocks? So then we step it up to another level with before-after control impact studies. And then in some other literature, they call them difference in difference studies, where we are measuring the trends in affected and unaffected unit. So say in this example, we look at mangrove cover, and we see that mangrove cover inside the MPA is maybe 1.2% times greater than in non-mangrove areas. This is a great approach because we assess trends as a result of intervention, we account for general shocks or changes, and it's seen as a gold standard for how we understand and measure impacts of interventions. However, we have to really think about what's our comparison sites. So say, for example, Chagos MPA, what would be a good comparison site to compare what is happening inside Chagos, a remote reef in the Indian Ocean? Do we have a comparable site to be able to account for shifts and trends? Or say in our example here, what if our MPA site was located way outside of a town where there's not much shrimp farming or other activities that may cause mangrove degradation. And then our control site was actually close to the population centers where it may be more subject to exploitation. So that is something that oftentimes is not considered. And in the case of MPAs and even Coral Reef MPAs, we realize that these are not randomly placed in the ocean. There's biases in where MPAs are. So if we were to say, what is the ideal scenario for measuring the impacts of marine protected areas, what would it be? Well, in other sciences, they would say you need a randomized control trial. So in taking this thought experiment to the ocean, it would mean taking all of our reefs, dividing it into equal grid cells, we completely wipe off all life off of the reefs, let it recover, randomly place MPAs in the ocean, and then measure the difference. So of course that is impossible and unethical in so many ways. And we have all kinds of challenges in the real world. Again, marine protected areas are not randomly placed in the ocean. There are different geopolitical contexts. There's then to really even measure and do reef monitoring is a very resource intensive exercise. And then there actually are a few studies or locations that collect information about what conditions were like before the MPA. This was picked up by Paul Ferraro and other authors. They looked at about 200 MPA studies and found that most studies are based on just spatial or temporal comparisons. They do not adequately control for some of the other factors that are important to control for understanding change. So what does this mean? Does this mean that we give up, that we can't really say for sure the effects of marine protected areas? Well, not so. There are other approaches we can use. And one we can draw from impact evaluation work. And what this is that we're compare trends in affected and almost equivalent non-affected units. And these sort of equivalent non-affected units is what we call a counterfactual. What would have happened at your reef at that point in time if the MPA was not there? And so in this case, if we had our storm before the intervention, if our control and our MPA sites are in similar location or in quite different locations, we would need to find a set of points or control sites that can represent a counterfactual, a comparative group, and then measure the difference. And we can say that that is a more accurate assessment of impacts. So the advantage of this approach is that it assesses trends or shift in trends that we can attribute to the intervention and accounts for general shocks or changes, and where we are comparing sites to a counterfactual, where we're sort of measuring apples to apples and oranges to oranges. And it kind of simulates a randomized control trial in that we remove the effect of bias in where the MPA site is. So I'll give you an example from some of our recent work where we attempted to do this approach. We obviously couldn't do it fully, and I'll explain that later. But we were interested in understanding the role of MPA management in shaping ecological outcomes on coral reefs and other habitats. We were interested in how MPAs are being managed, what are the impacts on fish populations, and then the linkage between the two. So we recognize that first we need to control for a few things. As I mentioned before, other studies have found that MPAs are often placed in where it's easy to place them, where there's less extractive uses and there's there's less political resistance, or in areas where there is high biodiversity in these tourism hotspots to protect it for tourism. And then we have a whole suite of other factors that affect fish populations based on ocean conditions, the social environment and human pressures, and then variations in space and time. And in our work, we realized that we did not have much information on pre-MPA conditions. So what did we try to do? So say if we have clip art reef over here, where we are interested in measuring the impact of this MPA, we may be tempted to just compare all fish populations that are inside this MPA to those on the outside. But obviously, there's lots of other factors we would need to account for here. What we really need to do is to measure the difference between the fish inside and outside locations that are similar to each other. Again, so we are comparing those apples to apples and oranges to oranges. And so we did this through a process called statistical matching, where we paired MPA and non-MPA locations that were as similar as possible and using statistical techniques to remove the remaining bias to then isolate the impact of the MPA itself. So the list of factors that we were trying to control for to account for the sort of opportunity costs, difference between locations or locations that may have high biodiversity value. We looked at distance to population centers as a measure of opportunity costs and habitat type and the distance to shore. And then for controlling for other factors, we ensured that the MPA and non-MPA sites were as similar as possible in terms of habitat, wave energy, and controlling for when samples and surveys were done. And to give you an example of what this looks like graphically, so before we match, this is the difference in the MPA and non-MPA sites from distance to population centers. And then after, where the one-to-one line indicates that they are balanced in terms of their distribution. And we did this using this data set, which represents a compilation of many different reef surveys as well as non-reef locations from around the world from a generous group of data providers, including AGRA, and we ended up with a data set of 15,000 sites and compared MPA to the statistically matched confactual locations. In the end, we had to remove quite a bit of data because we could not find comparable matches, but overall, we found that MPAs on average had positive impacts that were about 1.6 times greater, where MPA locations were 1.6 times more fish biomass than non-MPA locations, and most MPAs having that positive effect. We also found that those MPAs that had greater staff capacity and adequate staff capacity, those had greater impacts, almost over 3 times as much as MPAs that did not have adequate staff capacity, which was troubling because we also found that less than 10% of MPAs reported having adequate, which is the dark blue locations, staff capacity, and the majority, 90%, had none or inadequate levels of staff capacity. And so this was an important just talks about the importance of having adequate staff resources for management. So in summary, impact evaluation is a robust approach that we can use to isolate the impacts of interventions. However, it is very expensive to implement, to monitor before, after, and then some of the statistical approaches needed. And you need a really in-depth knowledge of the system to understand what are these factors, and that there are very few marine protected areas that actually have the criteria to meet this true impact evaluation. Nonetheless, it is an approach we can head towards as we improve monitoring design, compile different types of data, and draw on qualitative and quantitative approaches. Right, thank you very much. Sorry, we went over time, but if you have any questions, I'd be happy to answer.
Thank you, David. And for any questions, please put them in the Q&A for now, and we will answer them after Brigida's talk. Now, I would like to introduce to you our next speaker, which is Brigida van Tussenbroek. In 1990, Brigida started a seagrass research lab at UNAM on the Mexican Caribbean coast. She was pretty happy doing this research, but in 2015, her life as a seagrass researcher was, as she calls it, destroyed by sargassum. This is a brown algae that arrived on shores and led to the death of the seagrass beds she was actually studying. Since then, she has been studying the impacts of the sargassum brown tides on Caribbean coast systems, not just seagrass, and has become quite an expert in this field. And we are very happy to have you here to share your knowledge.
Thank you very much. Nice introduction, Francisca. I will start sharing my screen. I think you can all see it now. And I will be talking about the impact of the sargassum brown tides on Mexican Caribbean reef systems. It all started in 2011. When suddenly and unexpectedly satellite imagery picked up signals of algal masses in the southern Atlantic. And when they had a closer look, they appeared to be sargasso. Sargasso is polypelagic species of sargassum, Sargassum fluitans and Sargassum natans. And these algae started some years, there were more masses, some years there were less masses. But in July 2018, Wang et al. From Southport University reported an algal mass which extended from the African coast until well into the Gulf of Mexico. And they named this the Great Atlantic Sargasso or Sargassum Belt. And they estimated that the biomass of this bloom, this algal bloom, which you could call it, was over 20 million tons. So this is a different system from the original Sargasso Sea, which has always existed, which is more or less in the Bermuda Triangle. But the Sargassum presence in the Southern Atlantic Ocean is a completely new phenomenon. Actually, when Sargasso is in the open ocean, it's a really nice system. It offers shelter to many visitors, many organisms, juvenile fish, adult fish, many of them of commercial importance, turtles, and it even has its own endemic fauna. For example, what you can see here is a sargassum fish. But when it arrives to the shore, this golden tide, which they call it when it's in the open ocean, turns into a brown tide. And that's a completely different matter. So here's where our study area is situated in the Mexican Caribbean on the northeastern side of this great Atlantic Sargassum belt. And the Mexican coast is actually host a multi-million-dollar tourist industry. Those tourists come to see the really white beaches and the nice blue sea. So they're not really happy when they see a brown sea instead, due to the sargassum. And the Mexican coast is characterized by a fringing reef. So it's mainly bordered by a fringing reef. And it used to be very beautiful. This is what it looked like between 1989 when I arrived there and to 2014. Nice blue sea, reef in the end. And then the Sargassum brown tide came. And then afterwards, the system changed. Even if you can see here in this image of October 2019, the Sargassum is not there anymore, but the sea looks different. At least the near shore is sort of brownish. So the system changed. So, and that is something which I'm trying to tell in the next slides. So what happens during Sargassum brown tide? Well, there's these huge amounts of sargassum, of algae, which mount up the shore, start to die, start to decompose. This anaerobic decomposition releases hydrogen sulfide, which is actually a really nasty gas, and it can even be toxic when humans are chronically exposed to it. And then all the leaches of this decaying biomass gets into the near shore. And there it forms what Sargassum brown tide, which can extend for 10 until over 100 meters, depending on how much sargassum arrived, and the local setting, the hydrodynamic and wind conditions, etc. But basically, all this organic matter in the near shore, it blocks out the light, so photosynthesis is blocked. There's a lot of organic matter coming into the sea. So bacteria and decomposing bacteria just sucks up all the oxygen, and there's a lot of stress, and there's a high organic matter load into the sea. These Sargassum brown tides are not happening all the time. The first one in Mexico was reported 2015, which I'm not showing here, but we started measuring, and at least the people in my lab, team of my lab started to measure at weekly or 2 weekly intervals to collect biomass of sargassum just arrived on the beach and see if there were any temporal variation in that. And what you can see here in 2016 and 17, not very much arrived. In 2018 is a different matter. It started to come, and what you can see here above here, the biomass, if it gets above about approximately 20 wet kilograms per square meter, which is really very small area, then more or less we can expect a Sargassum brown tide actually to happen. And in 2018, it started in March with a small break, and then it went on from May until September. In 2019, it went on from April until September. What does explain this? Well, what you can see here on the bar below, this of course coincides when there is a lot of sargassum reported in the sea or not. So with the blue bars tells there was no sargassum in the sea, so there's no sargassum arriving to the shore. The pinkish and the brown is when there was sargassum or possibly sargassum in the sea due to measured with satellite images. And then we can see that sargassum also arrives to the shore, except for in Mexico, when there are no trade winds. The trade winds, which are more from southeastern direction, bring the sargassum from the Yucatan current, which runs parallel to the Yucatan coast, and it brings it to the shore. So for example, if it doesn't come from the trade wind, which is which you can see here with this reddish area, but if it's out there, outside there, even if there's a lot of sargassum in the ocean, it does not arrive to the shore. So there's a large interannual and a large seasonal variability in arriving of sargassum to our shores. This was how the coast looked like in the Mexican, along the Mexican. Mirador Nizuc is in the more or less northern Mexican. Puerto Morelos a bit more, a bit further south. And Shoal is, for example, is a site really very far south. And this is how it looked like before the sargassum brown tide. And this is how it looked like during the first sargassum brown tide reported in 2015. What we measured first was the oxygen went down. For example, in this graph in Puerto Morelos, you can see the upper graph is a normal oxygen pattern. And in the night, it's slow because no photosynthesis. It goes up during the day and down again in the night. But when there's a sargassum brown tide, it stays down low all the time. And this is a highly flushed system, so it doesn't really reach zero. But if we have other systems like Shoal, Xcalac, even Nizuc, you can see there are some coastal sections. This is from top. You can see this is the shore, and near shore there are some red patches, which indicates there's anoxia, no oxygen at all. And of course, if there's no oxygen, everything dies. So every fauna, every animal, which is unfortunate enough to be caught in this area of anoxia and can't escape anymore, they just die. Of course, the banthus, which is there, of course, has stress if it's not extreme, or also dies. And it also kills all the seagrasses, which are in the sun. And the lush, what you can see here is a lush Thalassia testulinum bed, which is the turtle grass, which is the dominant seagrass in the Caribbean. After that, it's just a sand plain with organic matter. And the seagrasses are of special interest, because the seagrasses, when they grow near the shore, they fix the sediments. And near the shore, which is the surf and the swash zones, the wave action really moves a lot of sand around. So it apports sand and it takes away sand, it moves it around, and usually during storms, there's an offshore transport. But if there are seagrasses in that zone, which is just the seagrass territory, this is the seagrasses first. The leaves attenuate the waves, so the wave action decreases. And then also the huge system of rhizomes and roots, which is really very dense, fixes the sand, and therefore they stabilize our beaches. But this is also the area which is affected by the sargassum brown tides. So as a consequence, if there's a sargassum brown tide, and the seagrasses are lost, as you can see here, before the sargassum brown tide, there's a Thalassia testulinum bed almost near the shore. This is near Cancun, Mexico. And after sargassum brown tide, you can see the first 50, 60 meters of all the Thalassia meadow is lost. And per consequence, also the beach, this was the blue line, is the beach profile before the sargassum brown tide. And the orange one is the one after the sargassum brown tide. And we can see that the beach at both transects 1 and 3, the beach diminished by 10 to 15 meters. This is the sort of photographic imaging for that. And you can see here the palm trees, the beach in front of that, the seagrass meadows almost until the shore. You can still see wave ripples in the far, but where there's seagrass meadow, it's very calm and the waters are very transparent, and there's well, not a really large beach, but there's a nice really beach. After the sargassum brown tide of 2015, in 2016, there's no seagrass anymore. They killed it all. The wave action is a lot higher near the shore. You can see the wave ripples. There's no, and also the beach is lost, and the sea is even eating at the roots of the palm trees, which were really far from the sea before that. Another consequence of the sargassum brown tide is an enormous input of organic carbon, nitrogen, and phosphorus into the system. For example, in Puerto Morelos in 2018, and Rodriguez Martinez with unpublished data collected from the hotels, it was estimated that approximately 48,000 cubic meters sargasso arrived per kilometer during that year, which corresponds with more than 1,000 tons of organic carbon per kilometer in that year, which corresponds with more than 40 tons of nitrogen and almost 3.5 tons of phosphorus per kilometer, which is 20 times as much as we already put in through contaminated groundwater. So this is an enormous input of organic material and nutrients. And of course, they don't disappear from one day to another. So for example, in 2016, 1 year after the 2015 sargasso brown tide, we went to measure light extinction coefficients in the sea, not only near the shore, just throughout the reef lagoon. And here you can see the results of 3 sites. And you can see that even 1 year afterwards, in the coast, in the mid-lagoon, and even near the reef, the water transparency is still a lot lower and expressed by a higher light extinction coefficient, which mainly means that the blue turquoise water, where you can see clear nice fish, becomes sort of greenish water, where really the colors of the fish are really not as nice anymore. Another potential contamination is a microbial community. Sargassum has its own microbial community, and of course, when it's decaying, there's again another microbial community associated with it. We really don't know the consequences of that. And then there is, sargassum also had a lot of toxic elements. Of main concern is, for example, the arsenic, and this is the total arsenic contents of samples of sargassum collected all along the Mexican coast. And a lot of them have about 40 parts per million arsenic, total arsenic contents, which is sort of legal measure of selling dry algal seaweeds as animal fodder. But it has a lot. We still really don't know what the consequences for the ecosystem are as yet. Another potential impact is that the sargassum, as I already told you before, in the open ocean, it has a lot of fauna associated with it. Some of this fauna is not native to the systems where it arrives to the coast. And for example, in a study in 2018, I think, well, it's published in 2019, we found quite a few animals associated with the sargassum which had not registered in Puerto Morelos before. And what it means, we really still don't know. They might just stay with the sargassum, they might not go into the system. Maybe they establish in the reef system, but they really don't become invasive. We really don't know yet, but it is a potential threat. Another potential impact is that it changes the trophic relationships. For example, Nancy Cabanillas de Ram and collaborators, they found that sea urchins, instead of feeding on the algae of the reef, they started feeding more on sargassum. So they're really changing all the trophic relationships. Again, what are the impacts in the end? We really don't know yet. The potential impacts on the reef, because this is a reef week, or global reef week. In 2018 and 2019, Alvarez Philip and collaborators, they really registered a really high incidence of the white syndrome or stony coral tissue loss disease. For example, a lot of corals, more than half of the live colonies were affected. And they related this high prevalence of the disease to loss of water quality. And most likely this loss of water quality was due to the enormous inputs of sargassum, but no direct relationship with the sargassum brown tides has been established until now. Another point is, which is a study by Antonio Martinez and collaborators, they found that the larval movements of Acropora are different when they're exposed to lixiviates of sargassum, to leeches of sargassum. For example, the larvae moved a lot slower, they moved not that far, and they started to move more in circles. And after a while, they stopped to move at all. So they really reduced larval displacement. And what the long-term impact of this is, for example, for coral connectivity, for genetic diversity, and for the health of the Acropora population at all, we really don't know yet. What we do know is, for example, that the seagrass community changes. For example, a large seagrass meadow, when it's in the influence of the sargassum brown tide, It first becomes a sand plain, but then when the sargassum is gone, it turns into an algal plain because they establish a lot faster than the seagrass meadows. And for the coral reef, when the wasting disease continues, etc., there's quite a large chance that it also converts into an algal reef. And what does this mean for our shores? So after sargassum brown tide, which I started in the beginning, the sargassum is gone, but the system is not the same as before the sargassum brown tide. There's beach erosion, there are algae instead of seagrasses, the reef is degraded, water is less transparent, there's a huge organic load, and we really don't know when this will disappear. We really don't know how long it takes to recover, but for example, a Thalassia testudinum population, the seagrass population, usually takes a couple of decades to recolonize fully again. The reef, we really don't know, but it most likely is decades. But that means only if the sargassum influx does not reoccur. And the patterns are that it continues to reoccur, which means that these changes will be permanent. What does it mean for our coast? Well, if we have a healthy coast and a healthy reef, the waves are breaking. There's a healthy seagrass meadow, which is attenuating the waves. They really decrease the undertow and sediment transport. If we have a sargassum brown tide, the rugosity of the reef is already decreasing a little bit. Higher wave action combined with deterioration of seagrass meadow, loss of seagrass meadows near shore, entonces there's a lot larger undertow, so the reef is a lot more exposed during storms. And if the future scenario, if we don't respond properly to this, then we lose the rugosity of the reef, we lose our seagrass meadows, and we get a really severe coastal erosion. So this is more or less what it happens. Now, the houses on the dunes are more or less okay. I think even in the present scenario, if we really have a significant storm or significant hurricane, there is already, with the coastal erosion we see now, there's already a chance that some buildings will be affected. But if we continue and we don't do anything about this, definitely we'll have a severe syndrome of coastal squeeze. So what can we do? Management of sargassum. Well, it's the sargassum bloom most likely is a human-induced phenomenon. It's most likely caused by a combination of factors due to global warming, which changes currents, which may have imported sargassum from the original Sargasso Sea into the southern Atlantic, causing this bloom. We really don't know. Or windage, it changed the wind regimes. It's also most likely the addition of nutrients from the sheds, the Congo, the Amazon, or Orinoco sheds, for example. And in addition to Sahara dust, all these together, human impacts, and they cause this algal to bloom. Sargassum, when it grows, it sucks up carbon from the atmosphere, and it sucks up nitrogen and phosphorus from the ocean, and it converts it into biomass, a lot of biomass. We already saw that more than 20 million tons during July 2018 in the ocean, and we already saw that it's more than 48 million tons cubic meter along the Mexican shore. So what can we do about it? Well, we can do nothing or do nothing or not enough. And then the sargassum is decaying, and then the nitrogen and the phosphorus and the carbon and everything else, it's released into the ocean, and it only exacerbates the bloom again. So it enhances the bloom. We get degraded coastal ecosystems, loss of ecosystem services, and in the end, degraded human well-being. The other management option is we capture, we retrieve, and we process. And that not only mitigates the causes of the bloom, it may even improve it. Because if you take out the sargassum, you take out the CO2 from global warming, you take out the nitrogen and phosphorus from the human eutrophication, so you may even make it cleaner. So, and we save our coastal ecosystems, and we preserve our services, and we maintain or even improve the human well-being. So again, the same principle. Sargassum captures ocean nutrients and CO2 in the air and brings it to our shores in a concentrated form. So if we retrieve and process the algal masses, which are destined to arrive to shore, not everything is destined to arrive to shore. A lot of it stays in the ocean and flows to the regional Sargasso Sea. But if we capture and process the masses which come to shore, these actions will not only save our coast, our tourist industry, our societies, generate jobs and incomes, but it will even also clean our oceans and our air. Just as a disclaimer, I really didn't mean to be this a very pessimistic story, because people think I'm really pessimistic, and at this moment it may seem desperate, but this is a really, really big event throughout the Caribbean and the African coast. It's man-made, and usually man-made impacts like global warming and eutrophication, the impacts are very gradual. They only are perceived by us scientists, which go over and monitor and monitor and monitor again, and we see subtle changes in the environment, which other people don't see. They even see fish, and they see a degraded coral, but they still see the colored fish. But the Sargasso and brown tides are so incredibly obvious. Their impact is so really direct. So if we are not even able to react as a community, international community, because this is a regional problem, and establish the right collaborations among government, society, and scientific community, I think it will be really difficult to have it for even more gradual impacts. I would really like to thank you for your attention. And thank to all the collaborators which participated in all the work we've been doing on Sargasso.
Thank you, Brigid. And I'm gonna go in and go to the question session. So the first question I have is for David. And the question is, just lost it.
Oh, sorry, I had typed an answer to the question.
Oh, you typed an answer already. But we can also do the answer live if you want to. So for sites with high amounts of marine debris interacting with the marine life, would you still consider those for MPAs? If not, is there future consideration?
Yeah, so that's a good question. And yes, if many MPAs do have marine debris problems from either and whether it be land-based or marine-based sources of pollution. And if we had a better understanding of the interactions between marine debris and conservation or management, then that would go a long way to understanding how we can preserve biodiversity with multiple threats.
Cool. And I got a second question for you, which you also already answered typing, but I would like to also hear it for the rest of the people who maybe didn't see the typed answer. And Sherade Bayana would like to ask, how much is the changing demographic of coastal populations considered when the evaluation of MPAs are conducted? Considering traditional communities tend to preserve their fishing grounds, while immigrants of people with commercial goals tend to neglect the interest of locals.
Yes, that is a huge and understudied issue in marine conservation. Just because coral reefs, like any other system, are coupled with human activities. And so the relationship between the people who depend on the reefs can determine the types of social and ecological outcomes that we see. So definitely an area that needs to be studied more, particularly migration and how that's led to negative social and ecological implications, and in some cases benefits. And so it's very context specific.
Yes. And I got a bit of a technical question for you. How do you calculate the counterfactual trend? And what are some of the specific factors you take into account? For example, fishing prior to the MPA, And how do you calculate it if there's not much data before the MPA was made?
Yes, and that was, I would say, one of the biggest limitations in our studies. The ideal setup is if we had an understanding of pre-MPA conditions from way before it was established all the way through to now, as well as the fish populations that are being fished. Those data, if they exist somewhere, please let me know. But instead, we used proxies for fishing pressure to match locations that had similar likelihood of being fished. So that will account for some, not all of the bias, but hopefully that can help to give a better estimate of the impacts of the MPA when you have imperfect data.
Yeah, and we never have perfect data as scientists anyways.
No, no.
I have a few questions for Brigitte as well. How can taking sargassum create income? How would people make money out of processing the sargassum? And Valerie would also like to know where people are actually doing this and managing. So it's in one.
Yes, that's a good point. Of course, taking out sargassum, yeah, ideally you should take it out before it arrives to the shore, because when it's already on the shore, the damage is already done, is of course expensive, which is one thing. So you ideally would have a processing and a revenue from the sargassum, whichever offsets the cost you already make with collecting it. But in this case, nobody has really done a decent business case, but you could include in that also the carbon credits, maybe, if people are doing it. And also, of course, the gain from the tourist industry, which is going to be destroyed now. So that's one thing for establishing good business case. Processing, there are many things you can do with sargassum, actually. People are using it until now as a fertilizer, which really needs to be further looked into. Now we know it's got a lot of arsenic. There's one really good business person here in Mexico, which actually makes bricks out of sargassum, which work really well, and they look very good, and they were professional. He's now building school even with those bricks. There are people making shoes out of them. You can use the alginates, and they are good for the pharmaceutical industry. Fucoidan, it has a lot of quite high quality fucoidan, which is also looked as an antioxidant, etc., in the pharmaceutical industry. It's been used as animal fodder, which again, I've some of my reservations. As you said, it has a lot of arsenic in it. And I think there are many other already practices. People are making paper out of it also. So there are many applications, and they still have to become incorporated in a business case, and you can use. And of course, one big, of course, which is one of the first logical applications of sargassum, of course, of any biomass is generating energy. So you can put it in a biodigester and generate biogas. It's not that easy, and sargassum has some properties which makes it difficult to do it straight away. You have to mix it, or you have to do pre-processing. People are still working on it, but there are many applications which are possible. One of the major challenges, and then I will stop the question, is of course that sargassum arrives irregularly. So you have to deal with, you have to build an industry with a primary source which is unpredictable. So you have to overcome that, which is an additional cost. It's not impossible, but it's an additional cost.
Yeah. As coral reef scientists, we often talk about
I'm sorry, Francisca, I don't hear the question.
Francisca, I
Okay.
think we lost you.
Okay. Are you doing it or shall I take the
Yeah,
question?
if you see one that you want to answer, you could. I think one question I see here from Diago Learman is asking whether or not the sargassum can be managed at the source. Are there ways to control the sargassum before it gets to the shoreline?
Well, the source is a huge oceanic area between Africa and Brazil. So it's really very big. And of course, when sargassum is at the open ocean, as I said, it's a system. It sucks up carbon, it sucks up nutrients, it's home to a lot of fauna and flora, etc. I don't see any reason why you should remove it from there. I think you should remove it when it's destined to get to shore. And that's maybe depending on the setting and where you are, it may be between 10 and 30% of the sargassum which is in the open ocean at the moment in front of any shore. So that ideally you should take away. And so it's attacking at the source. I think it's impossible. And yeah, just you're getting it before it arrives to shore. Of course, you need good imagery, you need good oceanic ground modeling, and you need the good equipment, etc. And this is a new phenomenon. It's an international phenomenon. So it has been, until now, it has been very difficult to coordinate this properly. But I hope it will help sooner or later. Well, I would really prefer sooner. This must happen to save our coastal ecosystems.
And I have another question for you, Brigida. Phil Karp was wondering, says that he's sure you're aware of the innovative initiative to ensure that coral reefs off the coast of Cancun are protected against loss due to storm damage. And do you know if there's any similar risk analysis being done looking at sargassum, or could a similar scheme be applied to mitigate the damage from sargassum?
It is very interesting, yes. I think we're really, it's a way we could look into it to get revenue to deal with this problem, because it's not cheap. That's something which is really very clear. It's not going to be cheap to capture it and transport it and process it. And there might be some revenue at the end, but the initial investment will be very large. So an insurance scheme similar to the reef, I think it's an option we could look into, but it needs to be done at an international level, because, as I say, it comes from it's not really one country, it's the whole region.
Thanks. And I see another question here for David, wondering if this is from Antonella Rivera, wondering if you're able to quantify the impact of distinct governance aspects within the MPAs, or if all the MPAs are being considered equal.
Yeah, so it's the distinct governance aspects within the MPA. Not sure if that means like different zones or the distinct governance between different MPAs. But if it is the zones, then yes, that's a further step that we're working on right now, where we are assessing the impacts of different types of zones within MPAs. And then the other question is yes as well. We have been trying to collect information on how coral reef MPAs in Indonesia are being governed, and in terms of the decision-making process and who's included, etc., and to see how that may be affecting outcomes. That's a good question.
Awesome, thank you. And let's just have one final question so we can try to end close to our hour window. Brigida, just wondering if you're seeing any positive impacts on the ecosystem of the sargassum. I know there's a lot of negative impacts that can feel a bit overwhelming, but wondering if there are any sort of positive impacts.
Well, as I said in the beginning, the golden tide, the sargassum in the open ocean, is a valuable ecosystem. And if it stayed there, it would be great. And for example, what has been seen, for example, is that these ocean masses are home to a lot of juvenile fish, which might find protection there from predation. And for example, the dorado or the mahi-mahi, wow, I don't know the scientific name at that moment, but it's a dolphin fish in English, is a really very nice fish to eat. And the fisheries, since the sargassum masses have been arriving, has increased. So if it stayed in the open ocean, it might actually help some fisheries resources, or it might change, but it might be positive. And of course, and as I said, it's a reaction to us changing our environment, the CO2 and the nutrients, and it tries to compensate for that sargassum, and it converts it into biomass. And so I think it sucks up a lot of carbon. It's a blue carbon. You can even consider it a blue carbon. It sucks up nutrients, so it cleans the ocean. So it's not all negative. It's just so difficult to deal with the part which arrives to the shores. But it's not all negative, of course.
Thank you. And I want to go ahead and say thank you to both our presenters today, Brigida and David. Thank you so much for your talks. And thank you to all the attendees who have been actively chatting and asking questions. Yeah, and here I just want to go ahead and remind everybody about what we have lined up for tomorrow. So yes, thanks again. And here are their contacts. If we didn't get to any of your questions, you might want to go ahead and email them your questions. And then for tomorrow, 3 to 4 Eastern Standard Time, we do have 2 more live plenary talks. So we'll have a talk on NASA grouper research in the Bahamas from Dr. Krista Sherman. And we'll also have a talk on the hidden habitats of the Caribbean depths from Dr. Diva Aman. And the talk from today, as well as all the other talks, will be available on the Global Coral Reef Week YouTube channel. So if you wanted to share what you heard today with somebody, it will be available there. That's it. Thank you.
Thank you.
Thank you very much. Thank you for attending. Thank you for organizing.
Thank you, everyone.